engaging video to Introduction to Microbiology for Unit 1 in nursing students. So I am Sornambiga, Assistant Professor, SNS College of Nursing. Today we are going into the topic of how this introduction to the microbiology, what are the insights are there, how we are going to learn about microorganisms and how they are reproductions, what are the microorganisms are there, how they are infected to their persons, human beings, and how we are going to take a treatment. These also we are going to learn from this video. So look at this, some of the colorful illustration microorganisms or invite to what actually inside is there. To the microscopic realm, it is a microorganism open the window of the bacteria to the prokaryotic powerhouse. To the simple cell lagging of true nucleus, but packed with it with efficiency. In this colorful is not a colorful, it is a dangerous one to approach and invisible also. Here, the picture of bacterial cell to compact cell sustain mission to roughly 1 to 10 micrometer in size to the FR and the cell capable to rapid multiplication to get a gel-like a fluid of hosting ribosomes, that is out protein of the nuclei and coiled with the DNA inside of that and region of protective nucleus surface. To the surroundings, in the membrane of the plasma membrane acting the selective gatekeeper to that for nutrition and to the waste materials also to excrete outside. And the cell wall present with peptidoglycan layer to present the shape of the bacteria. It gives the thicker for gram positive, thinner and gram negative cells to the lipids. And capsules also might be present in that way of immune cells to the cling of the surface to the urinary tract infection. In that best example is Escherichia coli to latches onto the bladder walls. In that motility, attachment of flagella with like a tails proportional to the cells to move one place to another place by the way of movement to exchange genetic materials also. Here in this bacterial structure, we saw phylus, capsules, cell wall, plasma membrane, and nucleotide like whether DNA or RNA, cytoplasms, ribosomes, flagellums. These are present in the bacterial cell wall. Then shape of the bacteria. How they are bacteria's shape. In that cocci appears sparse, solitary like a skin while with the diplococci, gonorrhea transmissions and chain up like a thread or cluster shape and that arrangement develops and rod shaped bacilli to stand tall, alone, diplo, strepto like of that in the chain like apparents and bite like a fevers and it is a gamma shaped bacteria. It's just in the gamma shape is there and the spiral shape was there. It is a spiral spirochetes are there and vibrio cholerae. These are the shapes of bacteria we are cocci, bacilli and spiral. So whether it is cocci is a one small round shape over there, it is known as cocci, and bacilli is one type of small rod shape is there, whether it is a positive or negative sense of bacteria. Here, gram staining process. If you are entitled with the gram staining process, first we are adding some crystal violet to the identification of bacteria until the 16 minutes. After we are washed with water, after that we are adding iodine solution. Iodine is one type of orange color solution. After the 16 minutes 6 seconds we are washed with water. After that we are decolorizing agent. It is a transparent solution, ethyl alcohol. It is decolorizing the solution from the gram positive bacteria. Whether it is lastly we are adding saffron in for 45 seconds. After that we are wash with water, tap water, and after that microscopic under the microscope we can able to see whether the gram positive bacteria are visible in the case of purple color and gram negative bacteria or whether it is visible by the pink in color. So we can able to easily identify which one is gram positive and which one is gram negative. Whether we are moving on to the viral structure, we saw that bacterial structure, how we are staining, what are the staining processes. So now we are moving on to the viral. The structure of elegant and minimal protein capsid encased with the genetic material, whether DNA or RNA. or single or double stranded also is there, while some of the then lipid envelope stabbed with the spikes to preferred with the infected cells for the camouflage and the entire to the camps of the SARS-CoV virus, crown with the spikes and lungs COVID virus, its jaws, and influenza virus, Zika viruses, life-threatening viruses, these are, and mono, nanoviruses, coronaviruses, rabies viruses, these are just the picornaviruses. These are we are not able to isolate from the laboratories. It is a separate laboratories are there to isolate the virus, and we are developing the vaccines also in the prime defenses. Moving on to this, you know very well about it is a fungal cell structure. In the fungal cell boosts the chitin walls unlike the bacteria and virus. In the mitochondria is also present and the DNA for the ribosomes and blooms also is there in that cases. Let's begin with the pathogens to the bacteria, virus, fungus and parasites host with the reservoir like a human carriers, animals, soil, food or water. So from there exist of the bioportal in the respiratory droplets, cough, sneeze, gastrointestinal fecus or else bloodstream brights or else urine and tears formation. These are the portal to spread one person to another person. By the way of transmission mode, it is a direct contact with the meter and airborne nuclei like tuberculosis and mosquitoes like malaria, typhoid, jaundice like this and splashes of that immunocompromised. These are the way we have to chain of infection one person to another person to sustainable host, portal of entry and mode of transmission to follow the proper hand hygiene methods. These are the way and the direct contact to spread the skin to skin to helps of that hands of surgical sites to wrap to close that airborne particles and indirectly vehicle contamination also and via foodborne pathogens, waterborne pathogens, bloodborne pathogens. These are the there two pathogens to spread the disease by the way of people one person to another persons. After that, how we are control these things? The control of the start sterilization to the including spores, viruses, disinfectant to essential aseptic conditions, and that these are physical method and chemical method of sterilization. Another physical method of sterilization, we have to follow three methods. One is a heat sterilization, radiation sterilization, filtration method. After this, we are following the dry heat method, moist heat method. Same like a chemical method also we are using gas and liquid method. So this flow chart hopefully will helpful for your examination purposes and your practical purposes also. And after that also some of the radiation method also we are using for the sterilization. Yeah, immunity. We are moving on that innate immunity and adaptive immunity. That innate immunity is naturally secreted in our body by the way of phagocytic process, and that adaptive immunity also we are induce the immunity by the way of B lymphocytes, T cells, and B cells. To the way of lymphocytes, it will be more effective to the risk factor to avoid the disease conditions. So this video will helpful for your learning purposes and your career purposes also. So thanks for watching.